Stem cells are those cells that reside within our body that have this capacity to repair and regenerate our tissues as we need. And they've been exploited in terms of cell therapies with a kind of an aim to replace tissues. So whether that's cartilage repair or the introduction of new islet cells into the pancreas to replace those insulin-producing cells lost in diabetes, that you would use a stem cell source to do. Stromal cells, on the other hand, also live within the tissues, but they're more there as a coordinator. And they're very good at communicating and talking to other cells within our body, especially the immune cells, and kind of orchestrating the responses that we have within our tissues.
Welcome to the Smart Biotech Scientist. I'm your host, David Brühlmann, and today we're diving deep into the challenging manufacturing of stromal cell therapy with an absolute powerhouse in the field. Meet Lindsay Davies, who's a consultant at CellTherEx supporting advanced therapy developers, and she's also a chief scientific officer at NextCell Pharma. And if this isn't enough, she recently elected vice president for Europe at the International Society of Cell and Gene Therapy. With over 20 years transforming how we think about stromal cell therapies, she is here to share game-changing insights that could revolutionize your bioprocessing approach. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Lindsay, to the Smart Biotech Scientist. It's a pleasure to have you on today.
Thank you for having me.
Absolutely. Lindsay, share something that you believe about ATMP development that most people disagree with.
I would say in the ATMP community, we're pretty aligned. We're all facing the challenges together. So it's a really unique field to actually be in and work with the community. But I would say that there's been a real drive to push manufacturing towards automated processes and things like that. And while that does offer advantages to a number of products, for many of us, the open process still remains the kind of the gold standard way to go at the second.
Lindsay, draw us into your story because you've had an incredible journey in advanced therapies. Tell us how you got started in this field, doing your PhD, and then what were some interesting pit stops along the way?
For me, I've always been someone who loves innovation, creation, puzzle solving, solution driven in whatever aspect that I've worked with. So I started very much in product design. And I was working as a designer and realized that, yeah, I love the innovation, I love the creation, but I really wanted to do something that was going to truly impact society and the quality of life of individuals. So moved into science, and my PhD was in cartilage repair, and that really excited me because I was working with sports injuries and that type of thing. And that was really my first introduction to advanced therapies and cell therapy, which was still very much in its infancy then. So it was really a novel field and a very small environment to be working within. I loved the basic research, but decided then to move to Sweden and to the Karolinska Institutet, as they were really pioneering clinical development of cell therapies, especially stromal cell therapies. So I moved there to learn about manufacturing, clinical trial development, how we could treat patients, how we could evaluate those patients. And I think from there, having like a real drive then to work with patients and society and make that impact, it was kind of a natural transition to go into industry and to really complete the product life cycle and then see how we commercialize these products and how we get them out on the market to make them, hopefully in the future, a standard line of care for everybody.
And in 2021, you made this shift from academia to the industry by founding actually several companies. Can you tell us a bit more what drove this pivot? What led you to finally not only going into the industry, but actually starting new companies?
I think working in somewhere like the Karolinska, which is really known for its innovation and what it can kick out into academic clinical trials and into industry and spin-outs. I was working with a lot of individuals who really were feeling those kind of bottlenecks and pitfalls transitioning out from an academic scientific environment into an environment where you're expected to understand business, investment strategy, and how to really make an idea into a corporate venture. I really liked that idea, so decided to leave academia and start CellTherEx, which is a consulting company that is focused on helping academics go through that transition and use both my own experiences, but my network as well to really give access to academics so that they can focus on what they're truly good at, which is the innovation and creation, and not make them have to be experts in everything, really giving them the support and infrastructure they needed to spin out from there. And I think it was kind of a bit of an opportunistic strategy to be able to do that, to really spot that gap. And QVance, which we co-founded as a subsidiary of NextCell Pharma, where I'm the chief scientific officer at the moment, was also opportunistic because we found through NextCell Pharma, as they were developing their mesenchymal stromal cell therapy for late-stage clinical trials, and we were looking at bringing manufacturing into Sweden potentially, that there was no quality control analytics company based inside the Nordics that specialized in cell and gene therapies. So QVance was really born out of seeing that gap in the market and real need for this specialist bespoke service. So I established that with NextCell Pharma to be able to really offer that opportunity to keep some of our products inside the Nordics, to reduce the amount of drug product that's being shipped all over Europe in order for QC testing, so that more of our product is actually going to the patient and not just to these tests.
As we're moving over now to the scientific and technical part of cell therapy, let's start with the scientific foundations, because there's so much going on in this space, a lot of new developments, a lot of different cell types. So what is the difference between stem cells and stromal cells, and why are stromal cells particularly promising for revolutionizing treatment of severe and incurable diseases?
I think there's a lot of confusion and cross-use of the nomenclature when it comes to stem and stromal cells. And that happens even within the industry with the kind of terms mesenchymal stem cell, mesenchymal stromal cell being used interchangeably. But when you look at the actual biology of the cells, it's for two very different purposes. Stem cells are those cells that reside within our body that have this capacity to repair and regenerate our tissues as we need. And they've been exploited in terms of cell therapies with a kind of an aim to replace tissues. So whether that's cartilage repair or the introduction of new islet cells into the pancreas to replace those insulin-producing cells lost in diabetes, that you would use a stem cell source to do. Stromal cells, on the other hand, also live within the tissues, but they're more there as a coordinator. And they're very good at communicating and talking to other cells within our body, especially the immune cells, and kind of orchestrating the responses that we have within our tissues. So there we use them for immune modulatory therapies. So stromal cells tend to be used more in the treatment of things like autoimmune diseases. They may be used in a severe infection. For example, they've been used extensively in COVID-19 treatment, or in things like reactions, rejection responses that you see, for example, in graft versus host disease in patients who've had hematopoietic stem cell transplantation. So two cells that are sometimes confused with each other, but have very different potential for developing cell therapies.
At NextCell, you're using stromal cells, if I'm correct. And tell us what kind of stromal cells you're using and what is the purpose for that and why are you focusing on that very cell type?
So at NextCell, we use umbilical cord tissue-derived mesenchymal stromal cells. And these cells have some particular advantages. We use them in the treatment of different autoimmune disorders. So our primary focus is type 1 diabetes, but we've also used them for viral pneumonia as well. So it's a bit of a platform technology for immune modulatory disorders. The reason we use umbilical cord is two reasons. One, we can use them allogeneically. That means that we don't need to match them with the patient. So we can harvest them from any individual and give them to any other individual. It's not like blood where we have to tissue type or anything. The other advantage is that mesenchymal stromal cells, they exist in most, if not all, of our connective tissues inside the body, but they have a very strong memory of where they've come from and what they were supposed to do in that location. So we can actually exploit that for how we want to clinically apply it. And with umbilical cord tissue, this comes from the baby and not from the mother. So these cells are very young, they're very potent, but they also are very acutely aware that the mother's immune cells are very different to the baby's immune cells. And so there we have this process in the body called fetal maternal tolerance, where they physically tolerate each other during pregnancy. And these cells remember that. So they're very good at modulating the immune system, dampening down inflammatory reactions, responding to things like infectious agents because they would be protecting the baby in the body. So there we can exploit them for a number of different potentials.
Now, how does the therapy work? For instance, you want to treat somebody with diabetes type 1. Do you need to produce the cells, quote unquote, in real time, or can you build them up and then store them for a long time? And then when the patient arrives, you can treat that patient, or how does that work?
So this is one of the amazing things about a mesenchymal stromal cell is that because it can be used for anybody without matching, it means that we can produce at scale, we can freeze the cells, and we can actually just deliver to the clinic when they are needed at the convenience of the patient, or if they are severely ill, then they're there and they're ready to go. They are thawed at the patient's bedside. They are mixed with just a salt solution, saline infusion bag, and then infused through the arm into the patient over 20 to 40 minutes. And that's it. And then if the patient, for example, has diabetes, we do this in an outpatient clinic, they can go home. There is no side effects and it's a one treatment. So it really doesn't impact their quality of life, or they have to have repeated treatments or monitoring by the clinics. So really good from that angle. How they work is, again, this is where they're different from a stem cell where you want to transplant the tissue and then you want it to stay there. With stromal cells, it has what we call a hit and run effect. So these cells, they enter the bloodstream and the majority of them will actually explode because they're not supposed to be in blood. And when they explode, they will release all types of chemical factors, cell membrane parts that are kind of eaten up then by our first line defense immune system. And that process causes those immune cells to become anti-inflammatory. And then we see this kind of domino knock-on effect where those cells then talk to our long-term immune adaptive cells and trigger an anti-inflammatory response there. And then in the case of type 1 diabetes, this is what then stops this auto-destructive response that we see through destruction of the pancreatic beta cells. So we know that like one infusion of these cells, the cells will be cleared from the patient within maybe 2 days. but the actual clinical effect that we see lasts at least 5 years.
Wow, that's amazing. And would that therapy then be, when you say 5 years, after 5 years, the patient would have to undergo another treatment, or in some cases that would be permanent?
That we don't know yet. We only have data from our patients for a 5-year follow-up. Our patients now are going into a longer 10-year follow-up to see just how long this effect can last and whether, as you said, this is permanent or whether this is temporary and we would give another treatment. What we do know is we have given a repeated treatment a year after the initial first treatment and can see that that can even potentially enhance the therapeutic effect even further by physically halting the disease rather than slowing the disease. So there's still a little bit more development that needed to be done, but the most important thing is that it's safe, it doesn't give the patient any side effects, and it doesn't require any kind of management or hospitalization or anything like that, or additional drugs needed to be used so that there's no side effects affecting the patient.
So you're using stromal cells to treat diabetes type 1, and we had this news in 2024 that there was a clinical trial, the world's first stem cell therapy that reverses diabetes. What are the outcomes of the two therapies? Are we getting similar results? Because as you mentioned in the beginning, the two cell types are very different.
Yeah, and this means two very different approaches. So the stem cell approach is actually to replace the pancreatic beta cells. So it's really targeting patients who have lost all of their insulin production. They have real difficulties in managing their blood glucose levels, and that makes them extremely susceptible to complications of the diabetic disease, so things like chronic kidney disease, cardiovascular disease, etc. The therapy that we're using is really targeting those patients who have been recently diagnosed. So they still have their own insulin production. It's just reduced and they need insulin support. There, we're actually halting the disease so that they can maintain over a long term their own insulin production without the need for stem cell therapy and kind of like other therapies you have to have alongside that for those stem cells to actually engraft and coexist in the body.
I'd like to dive a bit deeper into how you manufacture these cells, because you mentioned before, Lindsay, that you are able to produce at scale. Now, on this podcast, we have a lot of listeners working in biologics, and I want to stress one point, that biologics manufacturing is very different from cell therapy manufacturing, because now we are not using a protein as an end product anymore. Now the cell is the product, and this changes everything. So can you tell us how this paradigm shift changes the way you manufacture these new therapies?
Yeah, it is a challenge, especially as a lot of our guidelines and legislation for manufacturing and quality control are built off the back of biologics and traditional drugs. They don't fit inside the box very well, and therefore your manufacturing techniques and your quality control are bespoke to your individual product. And therefore, this kind of like interchange of ideas of what is acceptable really needs to be done at a very early stage with the regulators, so that you're both singing from the same hymn sheet, as we would say, in terms of how you're going to produce and what is acceptable to produce. How you actually manufacture is really dependent on the cell type, and their MSCs they're a challenging body to work with, I will be honest, because those cells, they truly control your process. Their biology is so strongly impacted by any change that you put into their environment that it makes things like moving from traditional 2D cell culture on plastic to a bioreactor extremely challenging, because things like shear force by putting them into like a spinoculture flask, or changing the topography of the surface they're on, or even the perfusion rate so that the exposure and the kind of communication with their environment changes by the movement of the media. All of these things impact their biology, and that impacts the quality of the product and how the product works. So control here is absolutely essential. And that means, I would say, as you're developing your product, simplicity is the key here, especially if you obviously want to tech transfer, you want to scale up, you want to move it to different locations, etc., etc. Reproducibility of a complex process becomes a real challenge to show robustness and reproducibility of that product across different environments where they're being manufactured. So there are many challenges there, but I think here is where the key is know your product, know what those cells are supposed to do, know what their critical quality parameters are so that you can check that continuously and make sure you are getting the same product again and again and again. And the other major issue is your starting materials. When you have biologics, this is all controlled in a very, not easy, but a very streamlined way that you can control. When you have different donor cells that will act in different ways, it becomes much more of a challenge because you don't have a standardized starting material to work from.
And that's a wrap for part 1 of our fascinating conversation with Lindsay Davies. We've explored her incredible journey and the fundamentals of stromal cell therapy. Don't miss part 2, where we will dive into manufacturing challenges and NextCell's groundbreaking work. Do you need help with your process development questions? Book a free call at brewmanconsulting.com/call, and I'm happy to guide you and help you start. And also, please leave a review on Apple Podcasts or whatever platform you found us on. I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.